without it sort of spoiling it for people who haven't joined yet, just give you a rundown of roughly what I'm going to say.
Okay.
I'm going to start with a little video that we helped the WWF, the World Wildlife Fund for Nature, put together, because they actually now are really into krill. It's one of their chosen species to support the conservation of. So they've got quite a lot of resources there to, help us put this material together to get to, other audiences beyond the scientific audiences, to interest them, and also alert them to some of the issues with krill and conservation. Then I'll just give you a little bit more
partnership with the WWF.
Yeah, so they have been quite supportive of scientists involved in our team and the wider team beyond the British Antarctic Survey as well, particularly early career researchers and students. And they go out, they actually, they meet philanthropists and other people who may be interested in conservation, may not, necessarily know about lesser known things like krill. Obviously everyone knows about pandas and tigers, but krill is off the radar. And so these people go out and go to forums and tell them about other things that the World Wildlife Fund would really want to support.
So it's really good they've got krill on their list then.
Yeah, it's been quite a coup for us to actually do that. It definitely needs support, and the more research we can do, the better, really. But yeah, anyway, it's a good example of the sort of thing they can do to sort of reach wider audiences. So I'll start off with that.
Right.
And then I'll give a little bit more science background. I won't go really into depth about that, but just to sort of give you an idea of why the particular thing that you're going to do when you're south is really interesting scientifically, so you can give a bit better context around the specific thing you're going to do. I'll play a few videos. Some of them you may have already seen, like the ones of Bjorg, the engineer at Bass, and myself talking about how to put the net together, but also the net working in the field. So I've got some videos of that as well. So that would be really useful for you to see. And then I'll go into maybe sort of the more boring parts of it about, what you have to do to actually treat the sample once you've collected it and things like that. I won't spend too much time. A lot of the notes that I'll be talking about are already in the things that I've consigned to the Bark Europa. So it's just to familiarize yourself with what you're going to find in the box, the various notes and sort of the ways to deploy things, the things you have to bear in mind, that sort of thing. Okay. So I'll run through that. I won't make a big deal of it, but just so for thoroughness, you're aware of what you're going to meet when you get there.
Great.
And that's it. I think the rest of it is just questions from you guys, really, about what you want to know. Maybe things I can't help you with at this point. I'll have to find out myself, but I'm here as a point of information as well. Good. I'm going to share my screen and have to sort of keep unsharing and sharing, because I'm going to sort of flip between videos and a PDF presentation. I'm not clever enough to integrate it all, I'm afraid. So it'll be a little bit disjointed, but I'll start off with the WWF thing. So I'll share my screen with that one.
If I may, in the meantime, I would like to introduce Matthew to you, who is one of the guides during your trip on board Europa. So you know.
Nice to meet you, Matthew.
Hello.
In the cold, deep waters of the Southern Ocean, a team of heroes assembles. Heroes unlike any you've seen before. If you don't know the name yet, now is the time. Krill. Antarctic krill. No bigger than your little finger, krill are superheroes of the Southern Ocean. They travel in huge swarms that can be seen from space. Their combined weight is similar to that of all the people on the planet. They could wrap themselves around the Earth 1,000,000 times. The biggest animal that has ever lived, the Antarctic blue whale, depends on krill. They even glow in the dark. Krill is too small a word. They are guardians of the underwater galaxy. Marvel at their superpowers. Krill feed the undersea world. Without krill, life for many other creatures in the oceans would be very, very different. And that's not all. Krill are climate heroes too. On a planet that's overheating, krill may have the power to help keep us cool. By shedding their exoskeleton every 10 to 14 days, and by eating plankton that absorb carbon dioxide, billions of krill are putting carbon where the sun doesn't shine. Millions of tons of it every year. That's why we all need the mighty krill. Now, more than ever. But even superheroes can be vulnerable. The climate crisis is krill's most fiendish enemy. It's turning these heroes into victims. Krill need cold water. Unfortunately, the way we have changed the Earth's climate means the oceans are heating, and the ice, which protects krill nurseries, is melting. And when krill are in danger, so is the web of life they support, and the whole climate of our planet. But it's a danger we can all help to fight. Even superheroes need assistance sometimes. Fighting the climate change crisis, working with nature, not against it, can help these superheroes of the Southern Ocean. There's so much we could learn from nature. Little things can make a huge difference. There's power in numbers. Heroes come in all shapes and sizes. Remember the name, the Mighty Krill.
There you go. Right. That's sort of taken us into a new league, really. I don't think I've ever seen such a big sell on krill before, but there we are. So, you know, there was a few things mentioned there, but one thing that you may have picked up on is that they were talking about krill nurseries and the fact that the conditions around these nurseries are changing quite dramatically at the moment. So there we go. Right. Okay. So hopefully you can now see something with 3 pictures of krill in various states of disrepair there. So these are some of the main features. They talked about molting. Molting is really important to krill because it's we're not quite sure why it does it so often, because most things like lobsters, they only molt once a year or even once when they're just about to become reproductive. Krill molt all the time, and all those molts go into the deep ocean. And that's why it's really important in terms of how they take carbon into the deep ocean. But the other thing I really want you to notice is how distended they become, how sort of large they become when they're in reproductive condition. And this was a poor animal that didn't survive when we caught it. So we dissected out this ovary, and you can see how big all of that mass that we cut out of it on the bottom right-hand side there are the eggs. And so over half its body weight of a female is actually put into reproduction to develop these oocytes, which they then lay around October, November time. So that's coming into springtime in the Antarctic. And what we're really interested in now is what happens next to those eggs, because they have a really unusual life cycle. So the eggs are actually negatively buoyant, they sink. And they sink for maybe about 10 days or so, where they go down if they're in open offshore waters. You can see the ones that are on the shelf on the left-hand side there. They're actually sort of being stopped from going any deeper. And actually those will struggle to survive actually because of everything on the bottom of the ocean that's going to try and eat those eggs. But if they're in the offshore waters, they can actually descend all the way down to 2,000 meters or so. Then they hatch successfully. And then it takes them about another 10 days or so to swim back to the surface again. So that's really quite an expensive way to spend your early part of your life cycle. But these eggs are full of fat, full of lipid, and that gives them enough energy to actually go through these early stages of their life cycle. But it's really crucial when they get back to the surface again that they're ending up in rich phytoplankton bloom so they can start feeding. And it's actually at that point that I'm hoping that you'll be able to catch these animals just as they're about to emerge from this sort of upward migration that they're making from their hatching depth. And these are what they will look like when you catch them. So you can see there's a scale bar there. They're not very big. It's 0.2 millimeter. They're about 1 millimeter long, these things. They can be slightly larger because krill is one of the largest of these. But these are the sort of early stages. They're called, it's not a very catchy name, it's called calyptopes. And they go through that period of their life cycle for about 2 to 3 weeks. They're developing all the time. Each stage that they go through, they molt, and then they sort of develop more limbs. They start to extend themselves. And then after about 3 weeks, they go through this major metamorphosis into a faucilia where they start to look more like a krill. That will, be a stage maybe you won't catch. But what I'm hoping is that you'll catch these Caloptopes stages. These are about maybe 3 or 4 weeks old, these animals at that stage. And this is really important part of the life cycle. It's so important that we try and model it. I'm not expecting you to pick up some of these details here, but it just shows how much effort we put in to actually understand what they're doing and try and work out the rules that allow them to be successful or not. So some of the rules that we think exist, if I can just talk you through, if you can see M1, there is a real limit to where they can spawn, so that this sort of reproductive stage, the females I just showed you, and actually where there's too much ice around, they can't actually spawn successfully. And actually, as I just mentioned to you, for M2, we think they have to hatch above the seafloor. So mostly where spawning will happen is going to be offshore, away from the shelf regions. If you go to M3, there's also a temperature which they have to be above in order to do this successfully. If it's too cold, they can't develop fast enough, and then they go too deep, and then they can't get back to the surface again. So -1 is about as cold as they can go. And actually, because they need to get back to the surface so quickly, the M4 part of it, starving, will occur if they're not in appropriate areas where there's enough sea ice to increase the productivity, but not too much sea ice. And then the other M5 and M6 are more about what happens so they can overwinter, because it takes them 2 or 3 years to become an adult. And this first year is probably the year where they have most mortality. So I don't expect you to remember all of that, but it's just to illustrate all these different parts of the life cycle that we're trying to parameterize for models, so we can actually make predictions about what might happen in the future if conditions change. That's why we build models like this. And this was just interesting just to sort of show you where you're going to be in the life cycle. So you're going to be there, so December, January time. And this, as I was just saying, is when the eggs and the Caloptopes are mostly going to be emerging. So that's the sort of stages. The Fertilia stages, these later stages, are going to be probably beyond the time that you're likely to be there. But that's the stage that you're going to be catching. That's one of the real critical stages. And this is a little bit complicated, but I'll work it through with you. This is actually just showing that what happens to the life cycle is that the animals are developing all the time, but they're not just plankton that drift around. They can actually move in their environment. They can make these migrations offshore and onshore, because actually they swim quite competently for a small animal. They can swim against some of the strongest currents in the Southern Ocean, in fact. So what happens in their life cycle is, if we just start at the bottom there, the adult population, if you see that bubble, what happens is that these adults are swimming to the left. They're swimming from the shelf region to the open ocean, and that's where they start spawning. And these eggs and larvae are then drifting in the Antarctic circumpolar current. That's the current you'll be fighting against as you're going south with the bark Europa. And they're drifting off with this current. But by the time they get to the winter point, they're actually then swallowed up by the ice, and that allows them to gradually move back inshore again when they become juveniles, and then they'll develop in that environment, which is a rich environment for them, until they become adults. So there's a horizontal migration, which is really important to understand as well, because you'll be coming in at certain parts of this where they're coming offshore, where the eggs are being spawned, and when the larvae are starting to drift. That's where you'll be intercepting these populations, I hope. And this is just a further background about why we're worried about krill. This is a paper that we published a couple of years ago in Nature. And this is actually showing eras of krill. So this is going back to loads and loads. We spend a long time digitizing old records of krill from sort of ancient expeditions, and then put them into a format where we can analyze. And if you see on the left-hand side there, the 1926 to 1939 era, actually there was a lot of exploration at that time. As you know, with Shackleton and everybody else, this was a rich period in exploration, but also some really meticulous science was carried out then. But in that era, when we put these catches together, we saw there was hot spots, the red areas there around South Georgia. That was a massive krill area. And if you go towards the peninsula, the Antarctic Peninsula, actually there wasn't so much krill around that area in that time. If If you skip forward then into the 1970s to 1990s, actually, it was quite evenly distributed, but you can see that there's a gradual increase in the amount of krill towards the peninsula. If you now go towards the modern day, 1996 to 2016, you can see that around South Georgia, the amount of krill has really decreased in relative terms to what it was in the past. Most of the concentration of krill now is around the Antarctic Peninsula. And this is a common phenomenon that we see in the natural environment with polar organisms, and actually mostly to do with the fact that their environment is warming up. So what they're trying to do is just to keep in the same temperature zone. They're having to follow these isotherms, we would call them, the sort of areas of equal temperature, further and further towards the poles. It's happening in the Arctic as well as in the Southern Ocean, and krill is conforming to this pattern quite nicely. It's not as straightforward as just following the latitudes, because there is sort of an east-west gradient as well because of the strong currents there and also the topography and everything else. But gradually it's actually more and more going towards the polar regions. And I'll show you another plot, which is actually in a paper that we're just bringing out in the next couple of months, which is actually comparing where the larvae are, that's the red areas, versus the blue areas, which are the post-larvae. We call them adults, juveniles, anything that's not a larvae. And you can see going through those same eras again, that if you just concentrate on the red areas, these nursery areas, the red areas were actually distributed just north of, that group of islands there are the South Orkneys. And so they're just north and east of there. But actually, if you go forward to the modern era, you can see that actually that area, that nursery area just to the north of the peninsula is decreasing. And now the main nursery area is much, much further down the Antarctic Peninsula. So it's retreating back. Now, the problem with this is that if he keeps doing that, it's going to come up against a big wall, because it can't go any further south than the Antarctic continent itself. So gradually it will find itself more and more marginalized if this is a pattern that will continue into the future. Anyway, these types of data are really important to us to keep collecting where larvae are, where they're developing, and actually seeing what its future fate is going to be. So that's why you're doing what you're doing, is actually trying to get more type samples for us to actually work out, versus the models, what we're predicting, what the observations are. That's what you'll be making. Right, I might stop there because I'm then going to go on to the practicalities. But if there's anything that you want to ask about krill biology or any other rationales that you want to be aware of, maybe this is a good time to ask.
What makes a good krill nursery? What are the features of a krill nursery?
Really good question. Yeah. The main thing is that it has a level of productivity that is sufficient for the animals to feed on. If you go out into sort of a lot of open ocean areas in the Southern Ocean, you find a lot of areas are not very productive at all. It's actually, it's called a high nutrient, low chlorophyll region. There's loads and loads of nutrients around, but there is a lack of a certain nutrient, iron, which stops phytoplankton from growing. If it's not enough around, and the phytoplankton can't bloom. So what you find, if you looked at sort of a big image of the Southern Ocean, you'll see little blobs of green and loads and loads of very blue regions. So the blobs of green are areas where there might be an outflow of iron, mostly coming from islands or peninsulas, and that's then seeding the area so that there's sufficient areas for phytoplankton to grow. And in much of the other parts of the Southern Ocean, it's just so far from these sources of iron that phytoplankton can't succeed at all. So where you find krill is where you find a lot of iron coming off and a lot of phytoplankton productivity. The other thing is that you need just about the right amount of ice. There's like a Goldilocks zone. Too much ice is bad for krill. Not enough ice is bad for krill. You just need these marginal ice zones, because there's sort of spaces between the ice where phytoplankton can thrive. But then you've got that ice that's actually generating a really stable environment for the phytoplankton to thrive in as well. So those are the conditions that we think are ideal. The other thing is that it can't be too warm. And we do a lot of studies around South Georgia, and there's loads of productivity around South Georgia, as you might imagine, because it's an island. There's lots of iron coming off it. There's huge phytoplankton blooms, but we think it's too warm there for these particular organisms to succeed. So it has to be the right temperature as well. But we don't completely know. You know, we wrote a paper a few years ago trying to model it, and our model was nowhere near great at predicting where we actually find larvae. So there's certain rules that we're still not aware of that we need to actually do further research on. So it's a very active area of research for us.
I was going to ask it, are there any visual clues above the surface that you are aware of? So, you know, are you predators or any seabirds or whatever that might indicate?
I can only talk about adults. We really don't know anything about larvae in terms of, you know, what their main predators are, you know, beyond what would normally be a plankton or like a mesopelagic fish or a jellyfish or something like that. But when you're talking about adults, when we've seen massive swarms, you see loads of whales, and that's really obvious.
Okay, yeah.
They will sort of I mean, some people have gone so far as to say they herd them up and almost farm them, move them around into areas where they know they're going to do better. You know, this is a bit fanciful if you ask me, but you know,
that's the adult krill rather.
Yeah, but for larvae, we really don't know. And at that point in their life cycle, they're not very good at swarming. Once they get to that fertilia stage that I mentioned, that's when they start to form the swarms that we regularly see in the adults. But at the calyptopy stage, they're just part of the plankton, like everything else, like lots of other small organisms.
Yeah.
So it's very hard to sort of pick out the sort of things that might be a giveaway for where the larvae might be.
Apart from, as you say, the amount of ice.
The ice is a really good indicator where we would expect to see them. If there's like a marginal amount of ice, that would be on average a good environment for the larvae to succeed in. Yeah. Good. There'll be time later on as well to ask questions, but what I'm going to do now is go into the net that we've shipped out for you. It's not like a modern net, as I would call it. This is the type of net that was actually used in those first expeditions I was talking about, you know, in the 1920s to 1930s. It's called a Nansen net. Nansen was a famous explorer who worked in the Arctic. He invented this net, and it has a number of really robust features. It's just a little bit too ancient for us to use now and to recreate. But what we did was that we faithfully recreated one of these nets because we wanted to calibrate what was done back then to what we would do with the modern net. So we tried to get absolutely everything, every detail exactly as it was described for this net that was used back then. Anyway, it's such a good net that we thought it would be appropriate for you. And also it gives you some connection back to, you know, the time of Shackleton and Mara as well. So that's what it looks like when it's dangled over the side. And you can see there's a number of meshes on there. They're not all the same going all the way down. It's all brass, which is beautiful. It's got a cod end there that we'll go into because that's quite important. That'll be the one thing that you really need to interact with quite well to make sure that works. You can see right at the bottom there's a weight just to keep everything taut as it goes into the water. I'm going to show you a video of what it's like when it's in the water, underneath the water. In fact, I'm going to have to stop sharing here and just go to that. So this is what you'll find when you open the boxes on the Bark Europa. So lots of different components, all looks a little bit puzzling, but we've tried to make it as self-explanatory as possible with various videos and also some help guides. But I think it's just seeing pictures of it, and Alan's got loads and loads of pictures of the nets that we took when we were putting it together for the demonstration that I'll show you in a minute. But just to give you an idea, not to be intimidated when you take this all out and think, how the hell does that go together? I love this picture. Just shows all the brass that goes into it. You know, we just don't get that these days. You know, everything's a little bit too technical, but just this old brass is completely indestructible, he says. Famous last words. I'll just show you another video then. Hang on, bear with me. So these are videos that I actually sent to Alan a while back, just talking about the net itself. We had it in our workshop and we just sort of talked through the net itself.
Hi, I'm Geraint. I'm a krill biologist, and you're lucky enough to be deploying our N70 net. Now, this is a net that actually goes back in history. James Marr actually used this net in his original krill expeditions to find krill larvae, which is what you're going to try and find for us at the British Antarctic Survey. So here's the net. It's a very old traditional design. There's a lot of brass involved, a lot of traditional materials, and we're going to run through how to actually put this net together. I've got somebody helping me here, who's Bjorg, and she's fantastic with these sort of things, so she will sort of explain to you each of the component parts. But essentially, you've got the mouth area here. This connects to the hauling rope. It goes down through a ring. You can see the net connected with a bit of rope. You've got some rough mesh here, and then this goes to a mesh which is 200 microns, okay, and that's really important. That's small enough to catch the larvae that we're after. And this runs all the way down to a cod end, which is here. You see how it connects to the cod end. And this is the part that you would actually unscrew with hopefully the sample in. Okay, and that would come out and then you deal with the sample. And this runs right to the end. And here is a weight to just hold the thing down. Okay, so that's the N70 net. We're going to run through each of these components in turn. Action.
Hi, I'm Bjorg. I'm just going to show you, run through the net, how you set it up. Starting at the bottom, which is the weight. It's sat in this holder, so when it's on deck you can put it into this, so it lifts out like that. Next thing is a swivel, so this turns around, coming into a shackle with 2 pieces of rope. This one here is just for holding the weight, and this other shorter rope is for attaching to the net. One thing to mention is with all of these shackles, including these galvanized ones and these brass ones, is to put either a cable tie or some seizing wire so that these don't get loose whilst it's in the water. Okay, moving up, we then move, there's these 3 wires collected into this one bigger shackle here. These again all needs to be tightened and seized. Coming then up into this holding ring and cod end. The ends of the rope will just slip through here, so you'll have to take the shackle off and then slip it through these rings and these rings. This is the cod end. Just looks like this. That screws into this upper bit here, like that. Make sure that is tight. The net then attaches to the cod end via a Jubilee clip. So Jubilee clip over the net, net goes onto the cod end, pushes back down, and then tighten the Jubilee clip so that you can't pull the net off. Right. Wires continue up the net like this, through this ring that will keep the net from twisting on itself. These then go all the way up to the top ring, again attached with a shackle, which will need to be either cable tied or seized. There are 3 holes for these 3 wires, and then the blue rope that was holding the weight is attached to this bottom, just under the rough net, again with a shackle, and this is then for hauling up the weight as you're hauling up the net. The net is attached to this upper ring via rope. Use the knots that you think is suitable, but trailing it around like we've done here is a good option, and then terminating it in the same place as you started. Further on then is these bars, which again are shackled onto the ring which the net is attached to. There are 3 of those, the same number as wires, coming into this top bit, which is the end and where your rope will go on for you to haul and lower the net in and out of the water. Again,
all of these shackles needs to be either cable tied or seized and properly tightened. Yeah, that's about it. Have fun.
Right, so that was a little bit maybe too detailed for you, but I suppose it's just to comfort you to the fact that there's lots and lots of explanatory information for when you're actually on the ship to actually put this thing together. There's also, you could see as Bjorg was describing that, there were sheets on the floor, sort of a guide of how to put the thing together. And as I said, we sent Alan loads and loads of pictures looking at each of the components when we put it together during that little thing that we put together for you there. The last video I'll show you. Let me just check there's no other details in the presentation. Hang on. It's just there's just a few more things about the just looking at it when it's all stretched out, I think that gives you much more information. But this is actually quite an important part of it about screwing that together. This is the thing that you'll be putting on and off all the time, so you'll get used to that. But it's absolutely essential that it's screwed on properly. The thing will vibrate a little bit when it's underwater. There's plenty of thread there to make sure that it doesn't come off completely, but just sort of double check with each other that that's going all the way in when you screw it in to put it for the next deployment. There's Will. He used to work in our team. He's actually doing something quite important there, which is actually rinsing the net off. He's just using seawater. I don't know what you'll have on the Bark Europa, whether that's possible. But what happens when these nets come out is that there's a little bit of zooplankton or even phytoplankton that's left on the net itself. And that's important that you're not contaminating the next deployment with what's gone before. I mean, it's good to be absolutely thorough, that you try and get as much off the net as possible into the cod end before you unscrew the cod end. But it doesn't matter if you miss a little bit, but the main thing is that you're not contaminating the next sample, because what we don't want to know, or don't want to sort of be misjudging, is where the larvae are when, you know, in fact, they're not there because they're actually from the last sample that you've taken. So that's quite an important step to do as you're leaving a station or everything else, to make sure this net is as clean as you can possibly make it. You might go through phytoplankton blooms and it could be quite green, and that's a bit of a pain, but do what you can to get everything off before you go on to the next station. I'll just show you a last video, if I can indulge you, of the of how they're actually deploying it on the ship itself. So this is actually something where the net's coming up, and you can see how the weight is coming over, and then they're taking the cod end off. Here we go. So you can see the cod end's just coming up there, and here's the weight that's trailing behind. So we've got the advantage of having a dirty great big crane on our boat, which you don't have the luxury of, unfortunately. This is going to be a lot of hand hauling for you, I imagine. So these guys have got it easy. You can see that guy there, he's actually then using a little rinse bottle. You've got these in your kit to actually give it a good rinse as well straight away if you want. They're pretty clean now. They're not so worried about rinsing it down because they can see there isn't that much gunk in the net. There's Will unscrewing the cod end. It's good to have a bucket nearby there. What you don't want to do is be sort of walking over the deck with that in your hand and then tripping over and it going everywhere. So if I was Will, I would have had a bucket right next to me to put that straight into a bucket. Don't leave it on the deck like he's done there, because you can kick that. I've done it too many times myself, so I know from experience how frustrating that is. Anyway, they've got more than one cod end, so they can screw another one back in and then go for a second deployment. So good to have somebody on the weight, making sure that gets over the gunnel. So they put the other cod end on it. Off he goes. Good. Okay, easy as that. Right. I think the rest of the stuff I've got for you now is just going into some of the more details about how to deal with the sample once you've got it up. So if there's any questions on the net itself, maybe this is a good time to ask before we go into how to deal with the sample.
With the rinsing down, Geraint, is it every deployment or every area?
Well,
they went to use it straight again then with a new cod end.
Yeah, they did that. I think they made a judgment call because it was very clean water. You could see that the net was really clean coming up, and they weren't going to another station. They were just staying there and doing the same thing again. So cross-contamination isn't something to worry about in that instance. If you want to get on with it really quickly, that's fine. However, as you leave, if you leave that station, you want to make sure that net is clean before you use it again.
Sense. And one more thing that while I thought about it was, are there any cable ties in that? Because she kept mentioning cable ties. Do we need to take some?
I don't think I got hold of them, I'm afraid. I know it was on my list.
We need to take some. Perhaps you could give us an idea of the size, because there's so many sizes.
Yeah. You could also use wire as well, use seizing wire, but that may be something that we only have on ships. But yeah, I'll try and sort that out.
No problem.
I think I might put them in. I can't quite remember. It was all back in the summer. Everything's a bit of a haze in COVID, really. It wasn't so easy to order stuff back then.
No, I'm sure. I just, if we need to take some, it's easy to take. So
I'll check. There's an inventory that I did send, so there maybe are on there. I can
there is. Yeah, I'll have a look. Yeah. Okay.
Right. I'll just carry on then with how to deal with a sample. Right, this is just so I'm not expecting you to read this, but this is actually something that we always do when we go to sea, is that we do these scientific equipment guidance notes about how to deploy it. So certain things to think about. This will be in your box, but things like the number of people you would need to do the deployment. It'd be good to have somebody with the radio to coordinate with the person who is actually in charge of the ship itself. Good to have just a seaman with you, and then three scientists, 3 scouts, whatever, to actually deal with the deployment. And I put 3 down here because I think actually because you're man hauling, you're going to need a little bit more oomph. There was just 2 doing it on that video, but actually they had a crane to help them, and you could see they did have a seaman with them just to make sure everything was safe. In terms of footwear, if you did have, steel toe caps, that would help. I don't think it's absolutely essential, but there are heavy weights being banded around. You're all going to have warm weather gear anyway. We said a hard hat because actually there's a crane overhead with us. That isn't going to necessarily be the case for you, and I don't think it's necessary unless you've got cranes dropping things on your heads. But if there is, things above your head, I would advise that there are some sort of measures to make sure it doesn't swing and hit you and make you unconscious. And things just to sort of, this is like a check of what to think about before you deploy it as well. Just making sure everything is steady, everything is secured, you've unlashed everything properly, and you're putting this down to 50 meters is what I hope. There'll be enough rope for it to go down to 50 meters before you start hauling in again. But it's going to be going vertically through the water, so hopefully it's just 50 meters of rope you need to get out beyond the gunwale to actually get to the depth you want. You won't know exactly what depth it's got to, because there might be a little bit of current veering it off, so it might not get to 50 meters. But as long as the standard is that you took 50 meters of water, 50 meters of rope into the water, then we know that's going to be standard throughout. And just about securing afterwards. So this is basically our, information to you to take that into account. It'd be good for everybody to read that. What we do when we do these sort of things is that we insist that everybody reads this before they get involved in the activity, so they're aware of all the hazards and things to think about. Okay, so these are a little sort of handy guide that I put together for you about how to preserve a sample. So after you've done the deployment, then I talked about having that bucket handy. There are buckets in your box. And to either put the cod end in the box or to pour it straight out in there. Once it's in a bucket, it's far more secure. You can see easy to kick these things over or to drop them. So as soon as you get into a bucket, the better. You'll have one of these handy squeezy bottles then to get all the little bits out of the cod end, because things will stick to the sides of it. Just we'll use as much seawater as you possibly can to get it out. Take that seawater from the same place that you've been at. Don't sort of carry it over from the previous station. So refill it at each station, because that's going to have the local plankton in there, because there will be contaminants in that water. I doubt you're going to get so much material that you're going to have to subdivide it, but this is just in case there's so much you can't get it into the jars that we've provided. So the way you would do that is, it's not sort of, it is bucket science basically. It's just sort of dividing it into equal halves, you know, as randomly as you possibly can. They may be into quarters and an eighths, but always make clear on your label that that's what you've done, so we know we're only looking at 1/8 of a sample rather than the whole of the sample. We can do the multiplication easy enough. And the reason we say to make sure you haven't got too much sample is if I just skip forward to 6, we don't want the sample to be more than 1/10 of the volume of the bottle, and the rest to be filled with the formal saline solution that I've given you. If it's not at this ratio, then the sample will not preserve properly, and we'll get sort of really badly half digested animals to try and identify. So that's why you need to sort of make sure that you've got just about enough animals to sort of, and you've got different sized bottles to choose from as well. But just make that sort of ready reckoning in your head about, you know, how much you need to make sure you got the appropriate amount. It's fine if it's actually a lot less than you might expect, and it's less than 1/10. That's fine, as long as there's sufficient, you know, formal saline to make sure that the preservation's happened. You've got a lovely sieve like this as well to concentrate the sample around, and a very important spoon to try and scoop it out. Try not to be too violent with that. Again, every time you scoop something, you're damaging something. So to do a sort of a combination of the spoon and the formal saline when you're getting the sample back out again. I've given you more than enough formal saline, probably for 3 expeditions rather than 1, so you're never going to run out of that. You've got loads and loads of labels. Put a pencil on that, not a pen, because the pen will dissolve. And actually, you've got all the boxes that you need as well, the official boxes that you can actually transport these samples in. They're UN boxes. Everything's come in these UN boxes, so all you're going to do is refill them back in again. You've also got the vermiculite. It's like a sort of a powdery substance that's inert. It stops any spillages from going to the outside of the box. There's loads and loads of layers. There's a bag in there as well, so it's impossible for the formal saline to leak out. It has to get through so many things, but that's necessary. We insist on all of that, because we don't want anything leaking or it being confiscated as a result of there being formal saline leaking out of the box. But that should all be fine there. Don't fill the box absolutely to the brim. Just make sure there's no more than 1/3 of the total volume of the box actually full of bottles. Again, I'm not sure you're going to be collecting so much that this is going to be an issue for you, but it's just to let you know. Okay, so that's how you go through the sample. I've sort of speeded through that. I only wished I could have taken you somewhere on a mini expedition to do this with you. I've tried to make it as self-explanatory as possible. You know, if it doesn't go to plan, that's fine. You know, as long as you've written down on the label, you know, what accidents happened or everything that, so we can actually piece it back together again, that's fine as well. You know, it's just that if we don't know what's happened, then we assume everything is fine, and that could be a bias then in our results. So just record what's happened so we can go back and just sort of know that that particular sample hasn't worked out in the way that we might have hoped. Good. There is actually this as well, which is a risk assessment about dealing with preservatives. Formaldehyde is not something you can swill about and drink. You know, it's not incredibly hazardous. This is actually diluted down, so it is transportable. It's not a sort of a regulated substance at that concentration. But as I say in this risk assessment, be good to wear protective gloves. I've given you loads and loads of gloves to have, you know, these latex gloves. You've got glasses and a lab coat, which would be really good to wear when you're doing this sort of transfer and using the formalin to top up the bottles. The bottles you've got are Nalgene bottles. They're plastic bottles, which we find are actually a lot better than the glass bottles of old. And you've got all the things you need to actually transport these samples safely. The rest is down to us about transfer and disposal. Good. So that's all the sort of the nitty-gritty of details that I think I can impart to you at this point. But as I said, the boxes, apart from potentially a lack of cable ties, should have everything you need in there, plus all those guides that I've just gone through. They're all been nicely sort of printed out for you and laminated and everything else, so they should be sort of seaworthy. Good. Right, so that's me done. I'm open for any questions you might have or any other thoughts.
Can I ask another question? I don't know if this has been discussed already, but when and where is the best place to do this? Are we doing, I don't know, this might almost be a question for Alan as much as you, Geraint, are we doing it every evening, or are we looking for specific places, or whenever we can?
I don't know your exact routes as yet, and probably that's something that may be still to be decided. There's no point looking for krill larvae if you're north of the polar front. It'd be good to do a practice haul maybe when you're in that environment where it doesn't really matter, but you have to get south of the polar front to catch Antarctic krill. So, you know, it's no point doing loads and loads of stations if that's a possibility for you when you're near Ushuaia or coming down, you know, through the Drake Passage that's north of the polar front. It is only really necessary below that. As soon as you're beyond the polar front, it'd be great to do it as often as you possibly can. You know, the more samples you get, the greater resolution we have. You know, so I don't know.
That time before is the perfect opportunity to familiarize ourselves with the kit, though. Yeah. Exactly.
We do loads of trial hauls before we do any in anger, actually. So you know, just when it doesn't really matter, that's the time to work out all the, you know, the little idiosyncrasies and everything else, and to get people familiar with the kit.
Okay, and the boat needs to be stationary at the times when we do this, doesn't it?
It does, yeah. I mean, you might have, it'd be impossible to keep it absolutely stationary unless you've got bow thrusters.
Yeah. So that's going to limit our opportunities, Lucy. Any other questions?
What are you going to do with the samples once we sent them back to you?
Yeah, well, we're going to analyze them. So down the microscope, I'll be because there won't just be krill larvae in there. In fact, there's definitely going to be loads and loads of things, and maybe not krill larvae in a lot of them. So there'll be things like copepods and chaetognaths, lots of other things which I hope you'll get a look at as well when you look down the microscope and put these animals under there and you see them swimming around. I've given you a little ID guide again. I think Alan's got that, you know, to actually try and work out, see if you can work out what these things are. But we'll be looking at the biodiversity in the catch. Even though krill larvae is the main thing we're after, you know, we're not going to ignore everything else that's in there as well. So we'll be going through all the species and the abundances and then comparing that biodiversity to our past records of that area. So at the end, you know, we will have a spreadsheet that you may want to see. I'll do lovely bar graphs for you with the things that you found. And look at, you know, how that compares to the wider context of, you know, what we find throughout the wider parts of the Southern Ocean. So whatever your catch is going to be scientifically useful, whether there's krill larvae in there or not.
So this is the zooplankton identification guide. So it's a 44-page guide, and it goes into great detail on all the different things that we might be able to see under the microscope, so that we can try and work out what it is we're looking at. So it's quite exciting. In terms of us looking at samples, how does that affect what we're giving to you? So do we need to look at separate to what we're giving to you, or do we?
Well, I did explain that if you want to do that, it's fine. You can divide the sample into two, and that won't make much difference to us at all, as long as we know you've done that. And you can take half and analyze it yourself and use it for what you want.
Right.
And just throw it overboard when you're finished, and you've preserved the other half.
Right.
Got it. No, you know, when you've got certain things that are so abundant, you know, if you split it by 50%, it's going to make no difference at all to us.
No.
There'll still be the potential.
Apart from the fact all the interesting stuff will be in your half.
Sorry, yeah, maybe if you see one big thing in my half, then that's tough, isn't it? Really.
Sorry, what would we be looking at the sample with on board?
So
we have a microscope. We've
donated a microscope to the Bark Europa permanently, so that is already on board, and so we've got use of that while we're on board. But then future expeditions might use it for other things.
It's a lovely microscope. It's one that we use all the time. That I recommended to Alan, and he got like a really good one off eBay or something, wasn't it? Oh.
It's Swiss, yeah, it's Swiss made. It was over £1,000 with all the bits. It's just really robust, high quality microscope. Pretty much unbreakable. I've said that now. I shouldn't have said that.
Yeah, we've got about 5 or 6 of them in our team, and we never let go of them because they're just so precious to us. We've taken loads of others, and they've just never been up to the top.
You can't buy them new now.
No.
Okay. Any other thoughts to share?
Yeah, I just wanted to ask, because you mentioned that ideally the net should be at 50 meters, but that we can't know that for sure. How much does it matter if it's not at 50 meters? And if it does matter, should we be measuring the depth with some sort of pressure sensor, ideally, or something? I don't.
Yeah, absolutely. You're quite right there, George. The thing is, when we write up our protocol, we actually make allowance to the fact that we don't know, beyond the 50 meters of rope out, that it's gone to 50 meters, and so that's like a known error for us. If you had any pressure sensor that you can put on the net, that's brilliant, but we didn't have one that we could offer you that would be easily deployable. So we are just going on the assumption that, whatever depth it's got to, it's going to be the upper part of the water column. What's really important to know, I suppose, is that when you're sampling above 50 meters, you're mostly sampling in what we call the upper mixed layer. So there's various strata within the ocean, but the top 50 or so, or 75 meters, depending on where you are, the world's oceans, that's almost like a washing machine because of the action of waves and winds. It just stirs everything up at this top strata. So if you only get 25 meters down or 50 meters down, you're probably going to be catching the same thing. It's not as if you're going to go through a big layer and then everything is different beneath that. In the top 50 meters in the Southern Ocean, it's not going to make much difference whether you got to 20 meters or 50 meters. It's good to sort of have a good sample, which is why I'm asking for 50 meters of rope to get out there, so you've got like a good catch coming back again. You're in the water for a sufficient amount of time to catch things, but whether you make it to 50 meters it's nice to know exactly where you went, but it's not as vital as you might think.
And what we've said right from the start is even a few samples is still useful, isn't it?
Absolutely, yeah. So, it's great to
get as many as we comfortably can, but obviously we need to be conscious, of other people on the voyage and respect for that and so on. But it's a case of if we get the opportunity, we'll get as many samples as we sensibly can. Yeah. Can I, on behalf of the project, just say a huge thank you for everything that Bass have done and lent us for this project. We hope we can get some samples for you.
So I wish you the best of luck. I'm very jealous. I'm not going south this year, so yeah, you'll be doing it for us. So yeah, I wish you all the best.
Okay, if anyone thinks of anything else after the then we can get an email to Geraint. So if you sort of wake up in the middle of the night thinking about krill, then drop us an email. But I think we'll let Geraint get on. But thank you so much, everyone, for attending. And I'll try and edit this so that we haven't got the second video playing. And what I'll try and do, Geraint, is pop the actual videos in where you played them, so that they'll go at full speed for people. Yeah. Okay. Thank you, everyone.
Right.
Bye.
Okay.
Cheers.
Take
Thank
care.
you.
Good luck.
Thank you.